Okay, thank you. Hello, everyone. My name is Weicai, and I'm coming from HKUST Guangzhou. So I'm very honored to present work MutBERT, Probabilistic Genome Representation Improves Genomics Foundation Models. Okay. So let's introduce the genome foundation model. And it's a language model that trains on raw DNA or RNA sequence. And it learns the language, the knowledge in the genome language. and it will be applied to downstream tasks. For example, functional region identification and disease SNP predictions. So over the past few years, large language models have revolutionized not only NLP, but also genomics. We can see models like the MutBERT and the nuclear transformer tackling complex tasks in functional genomics. So source from NLP, functional genomic foundation models also adopt similar design ideas. They tend to increase model size and incorporate diverse data size to enhance the model performance. For example, the first version of nuclear transformer, it has 4 types of model size from 50 million parameters to 2,500 million parameters. In addition, both the MutBERT and the second version of nuclear transformer, they all use data from multi-species. So what's the difference between natural language and the genome language? Okay, the biggest difference is the information. So the last line, especially natural language has dense information and genome language has sparse information. So in genome language, single nuclear variants can drastically change functions. So it has very sparse information. And the main challenge in genomic foundation model when we apply LLMs to function genomes is the redundancy. It limits the data efficiency, such as SNPs in 1,000 genome project. Okay, think about we have n samples, and given the specific region, the most sequence are almost the same, only some mutations. So we can call this mutation as SNPs, single nuclear variants. And these SNPs are very rare, but very important. They have something with, for example, disease association. So from this paper, we can see the redundancy in 1,000 genome project. SNPs only accounts for only 0.70%. In other words, expect for these variant regions, most sequences are the same, regardless of how many people's data we use. So for traditional methods, we have to loop the same sequence again and again to learn the knowledge in the genome language. So the main challenge, so the learning strategy is very redundant strategy because we have to loop the same sequence again and again, and this will be will become more challenging when we move to larger data sets such as biobank data. So what we need is a way to focus on informative variants without sacrificing sequence coverage or biological fidelity. And we train 3 models on 3 different data sets. And we train a baseline model on human reference genome. And for the mutation data size, 1,000 genome project and multi-species data size, the only difference, well, the 2 differences are number of SNPs and the number of samples of SNPs. For 1,000 genome project, it has about 3,000 people with rare mutations, but for multi-species data set, it has about 100 different species with many mutations. But these mutations may be less representation for people, and I will explain later. So this page shows the details how we construct the mutation data set. Firstly, we need to download the data from 1,000 genome project and multi-species data sets. And after that, we can get the detailed information by using BCF tools. For example, the name of a chromosome, position, reference, and alternative base pair, and the corresponding probability. And then we can split the whole human genome by base pair of n, and we will skip the segments with too short lengths. So as we can see the position information, and after that we can use the position information to align the segments because we split the whole genome by base pair of n. We can apply the segments with the detailed information in the table, and finally we can get probabilistic matrix. The detail is we can convert the raw input, raw segments into a integer input, and then we can use some function to convert it into a one-hot matrix, and then we can modify the information into the matrix to get the probabilistic matrix. So based on this, we propose probabilistic genome representation. And the key ideas are masked language model embedding. So we can start with unlabeled DNA sequence. So given the reference sequence, we have the alternative sequence and the corresponding probability. For example, the second position, which means we have 10% that T will mutate into G, and we can get the probabilistic matrix based on this information. For example, if there is no mutation and the vector will be a one-hot vector, otherwise a probabilistic vector. So we can achieve a masked matrix by using masked language modeling, which means each vector have a masked ratio that that be replaced by a masked input, and we can get the probabilistic genome representation by matrix multiplication. Detailedly, we can perform the matrix multiplication between masked matrix and the embedding weights. So for example, the input embedding is a weighted embedding, and it equals embedding A times 80% plus embedding G times 20%. So here is our architecture, and we named it MutBERT. It's a masked language model that trained on just the human genome, and it takes in probabilistic input instead of a single sequence. And our input is probabilistic genome representation, and we integrate, for example, flash attention and rotary position encoding into the model to enable longer context sequence. And we also modify the loss function. So the model can predict one target if there's no mutation, otherwise two targets. And this page shows the details of our loss function. So given the raw sequence input, we have the reference and the alternative base pair. For example, A, here A is mutation. And then we can get the masked input by using masked language modeling, and we can fed the masked input into the MutBERT to get the predictions. Here we have two cases. Case one, no mutation. So the label will be a one-hot label, and the loss equals to, for example, prediction of T times 1. And for the mutation case, the label will be a distribution, or we can say a probabilistic vector, and the loss will be a weighted loss. For example, it equals to prediction A times 80% plus prediction G times 20%. And it should be noted that the true probability may be too small because the mutation may have a low probability. So we add a temperature and a softmax to enlarge the probability. So our model can learn the mutation in the genome language, and to learn the knowledge. And we compare our MutBERT with other sorter models such as DMBERT2 and the nuclear transformer. And our model are significantly smaller and trained on field tokens, as we can see. And for the and we make a file comparison to focus on our method because all three MutBERT series have the same settings. So we evaluate our models on multiple downstream tasks spanning transcription, factor binding site prediction, file data size, and NT downstream tasks 18 data size and EQTL prediction. And it has 3 distant bins. And for tasks with red boxes, MutBERT series have to scale the model length from 500 to 1,000 for NT downstream tasks and 2,000 for EQTL prediction tasks. And the metrics used are MCC, MCC, and AURC respectively. So here is the performance, the overall results on 23 data sets. And as we can see, MutBERT outperforms most of the models such as DMBERT2 and most type of nuclear transformer. And it ranks third, while among all models, only behind the nuclear transformer with 500 million parameters, which was trained on multi-spaces, and the nuclear transformer, the first version, with 2,500 million parameters, which are much larger than DMBERT2. And for the internal comparison, MutBERT, it was trained on 1,000 genome project. It ranks first, and then MutBERT multi and the MutBERT reference. So this means probabilistic inputs are more informative than deterministic inputs, and due to the usage of multi-spaces, but it may introduce the noise because it compiles the difference between human genome and other different species such as mouse and bird. So it may introduce the noise, and the mutation is not independent. So MutBERT multi underperforms MutBERT. And in TFBS tasks, the model's performance varies. The lowest score comes from nuclear transformer with 500 million parameters, which was trained on human genome. But in construct, MutBERT achieved the best performance with the same human data. And from the comparison of 2 versions of a nuclear transformer, we can see large models usually have a better performance, and this phenomenon will be more obvious after using multi-spaces. And we can note that DMBERT2 achieved the second place, even though it used the multi-spaces data slightly lower than MutBERT. And in NT downstream tasks, the model's performance are all good, all exceeding 63, and the lowest score comes from DMBERT2, and the highest score comes from the NT V2 with 500 million parameters, which was trained on multi-spaces. And MutBERT ranks third, only behind the aforementioned models such as NT 2,500 parameters and NT V2 with 500 million parameters. And we can compare the aforementioned model, and we can see this illustrates the importance of technical innovation because for the second version of nuclear transformer, it has some technical improvements, so they can get a better performance, even though it has a smaller model size. And in EQTL prediction, actually the scores are almost close. The score range is from 0.590 to 0.614. And despite this, MutBERT also achieved the best performance, which means MutBERT is more sensitive to SNPs. And MutBERT multi also achieved the similar performance, which means it is more sensitive to SNPs because it uses multi-species data. So in conclusion, we propose MutBERT, a lightweight and efficient model that leveraging probabilistic genome optimization to enhance the model performance, and it achieved robust model performance by comparison with other models. And in the future, we can scale the model size up and incorporate multi-space data set to enhance the model performance. And we also can explore deeper variant and covariant modeling to improve the performance. And this work cannot be possible without these people. So thanks to my colleagues. Okay, thank you all.
Yeah, thanks for the talk. Questions?
A great talk. I have a simple question. I see the probabilistic matrix is built based on the allele frequencies, and I was wondering, the allele frequency was very varied for each pair, for each base across different populations. So how can model like be generalized to other populations or in this scenario?
Well, yeah, the allele frequency are usually the rare, and the probability usually small. So we modify the loss function to enlarge the probability. So we input the probability. We modify the probability to with a data augmentation, and we and a temperature, and we also use the softmax function to enlarge the probability, so the model can learn the knowledge in the genome language of the mutations.
Okay, hi. My question was, if you scale, like, I guess your results might improve if you scale up the model and maybe add more data, but in that case where you didn't do that, did you test how much, for example, one downstream task such as variant effect prediction improves as you have a smaller model and then scale it up to the size that you presented here?
Sorry, I'm not listening very clear.
Did you scale down your model and then did an ablation where you scale it up again, downstream tasks improve?
Well, we did not scale up the model size, and this is the future direction. So in the future, if we scaled up the model size, we can get a better performance. And we also want to incorporate diverse data sets to enhance the model performance. But maybe in other methods, because we integrate multi-species mutation into the input, but we did not get a better result. So maybe we can try other methods.
Yeah, thanks.
Thank you.
Yeah, yes, thank you again.